Welcome to Prescribed by Dr. Nathan Goodyear, where integrative oncology meets holistic healing. This podcast is your home for the latest in innovation, exploration, and elevation in all areas of integrative oncology, medicine, health, wellness, and beyond. Join me and a host of other innovators as we go from practicing to prescribing. Your daily dose of wellness begins now. Welcome back to another Prescribed by Dr. Nathan Goodyear. We are going to dive into a very contentious topic here. And as I've said, this new rebranding of the podcast, we're not going to leave any stone unturned. We're not going to leave any question unasked. I don't care how politically opposed there are those out there. We are going to focus on the science, follow the science to empower you with knowledge to grab a hold of your healthcare and steer it the way you want to. And so today I'm bringing back on a guest that I've had on my previous podcast, John Catanzaro. John, welcome to the Prescribed by Dr. Nathan Goodyear podcast.
Thanks, Nathan. Thank you for having me on again. Appreciate it.
So John and I go back a few years, and I'm going to let him talk about his area of expertise and knowledge as it relates to this. But this topic that we're touching on here, and you'll see the connection with John in just a moment, it's turbo cancer. Now, we live in a world right now where we're supposed to have a pronoun for everything. We can't describe something that we see. See, as a practitioner, what I've seen in cancer is cancer that has changed. I've seen cancer that has behaved more aggressively pre-2019 to post-2019. But as the great Orwellian statement from 1984 said, we're told to ignore our eyes and our ears. But what we see is a cancer that behaves turbocharged. Now, from our perspective, if we speak about turbo cancer, that's not allowed. It's fact-checked. But if we look actually at the conventional literature, these are quotes from published mainstream journals. Turbocharging chemotherapy, quote, end quote. Turbocharging the T cells to fight cancer, quote, end quote. And finally, race to supercharge cancer-fighting T cells. See, turbocharging is a term that is accepted according to a central narrative. But if you're outside of that central approved narrative and we say, hey, we see cancer behaving very differently pre-COVID and post-COVID, that's not acceptable in that narrative. But what we're going to do is we're going to bring in an expert in this area of really peptides. And I'm going to let him just kind of take over in just a moment, because I want you to understand the implications of why we are seeing turbo cancer, but where it's going. What does the future hold? Because we in our human nature, we tend to be very selfish, focused on ourselves. But what's the legacy that we're leaving? When people look at government, they think, wow, our government's leaving a legacy of debt. What if our government is leaving a legacy of something much, much graver, much grimmer? And that's cancer and other disease and an increase in early mortality. And that's where we're going in this podcast. John, your history, what you just tell the audience, tell the audience your history, because it is diverse as they come. And it's that diversity that I think puts you front and center in this arena, and I think provides the expertise.
Sure. I started, you remember, Nathan, I started in the aerospace industry, and then I was in there with sophisticated technology in the Air Force and then at Boeing. And then I went to medical school, the Bastyr University. I decided to go through more integrative training, more naturopathic type medicine approaches. I went through their program there, and I graduated back in 95 and then entered practice in 96. And then from 96 on all the way through 2000 and, around 2017, all those years were filled with practice and seeing very resistant cancer types, right? So very advanced cases, and had a large center there outside of Seattle. We had 8 physicians, and we had a research lab, and we were going gangbusters, seeing so many different complicated cases, and I was getting referrals from everywhere, not just in the United States domestically, but I would be getting patients from internationally. And we were making a mark basically by challenging the system, so to speak. So it wasn't just about practicing integrative medicine; it was about practicing innovative medicine. And through these resistant cancers, I already knew what the integrative modalities were, but I also knew that there were limitations, and there was no possible way you were going to break the spell of the cancer and the resistance, right? So challenged me to really go outside of the norm in the standards and begin the process of in more surveillance and interpretive type medicine. So the thing is that I started getting very engrafted in molecular and proteomic expressions in the disease patterns of the individuals that would come in with different types of cancers, including breast, prostate, sarcomas, pancreatic cancer, as we talked about just a few minutes ago, all types of cancers, blood dyscrasias, etc. And so got to learn those patterns. We got to see early on the developing patterns of resistance in these cancers and the certain gene protein relationships that came, and the variances associated with them, right? So no two people are alike. You got to see maybe the same diagnosis, but with a different expression. And so then we engineered personalized peptide therapeutics towards those bad aberrant pathways. And I would say 7 out of 10 times we were hitting out of the park and getting patients to go into a complete remission pattern and a disease-free survival over time, to where we have patients that are 14 years plus now, that were treated under old generation. Now, Neo7 Bioscience came into existence in 2019, before COVID started, and we assembled the whole technology platform, and our team developed, and we put all these pieces together of a molecular surveillance, interpretive mapping, and modeling type of a system that allows us to grab so much molecular data. It's impossible for a human brain to understand how to assimilate all that, and create a high definition engineering and in an autonomous environment, right? So it actually, instead of us trying to figure out how's that going to work, it's all in an engineering platform that allows us to design specifically for those aberrant molecular pathways that are resistant, creating more cancer, right? And what we're finding is we're getting much more improvement, because we're learning for each data set that comes in on a specific cancer type. We get to learn more about why those resistant patterns are present, what the relational crosstalk ability is. Like, in other words, how do these primary targets communicate with downstream targets, what the relationship of those targets are. And then we really fine tune the multi-targeted motif, right? So you already know that cancer is not a single target expression. It's a multi-target expression. And a lot of the times, downstream effectors are even more essential than the most fashionable targets that everybody knows about in conventional medicine that seem to burn out very quickly, right? So it's like you can focus in on these for a time, but there's no guarantee that they won't eventually smolder again, because it's just the nature of the landscape, right? So we have to get better at being able to know how to navigate and triage and allow for better action, right? So that's what Neo7 Bioscience does, is that we've put together that type of a architecture And we have a lot of designs that we've already done since 2019. Now, the clinical part of it was developed with testing over 500 patients early on in generation one. And so we got to lay down a lot of significant meat and potatoes, if you will, of the process by coordinating with high-end labs on the East Coast. We did private contracting of our labs partners at Dana-Farber Cancer very early on, and we worked with them for several years. So I would say from 2001 to 2014, we were laying down a lot of technology data. And then we took those data sets and brought them into the new engineering at Neo7 Bioscience and went through. Now we're going through we have a four generations of improvement in our iterations on our platform, right? So we have an active disease platform and we have a resilience platform. And we're working in both of those areas with many, not just cancer now. We also have neurodegenerative. We have autoimmune expressions, chronic infections, things like long-term, post-sequelae SARS-CoV-2, which is considered to be long COVID, with mRNA vaccine related complications that we're seeing a lot of. And so that pretty much gives you the little synopsis of how this thing has come about,
And I've known you for a few years. And, when we first met, it was that diversity, because I look for people that are not, mono or singularity in their thinking. They have a background that is diverse, because it's that diverse background that, that I think fuels a curiosity mind to provide different solutions to complex problems. And the problem we have in medicine today, particularly in cancer, is we have a monolithic form of thinking. It's very much an echo chamber instead of a curiosity of mind to look at complex problems and provide new solutions. It's like that old adage, how many engineers does it take to change a light bulb? It might be, well, how many doctors does it take, to treat an infectious disease? It's obviously not a joke there because of the past 5 years, but it's a reality. But that's what groupthink has gotten us. And we need more people like you working with other people like you to actually help us to bring forth this new frontier, I think, in medicine and precision cancer care. I love that, the right patient for the right treatment for the right patient at the right time. And, I've talked a lot about turbo cancer as it relates to, what we see in the clinical setting. And I've talked about it as it relates to the spike protein in the process and what I call the metastatic map. I also talk about how, if you look at the conventional literature, they describe, 8 different oncoviruses, cancer causing viruses, hepatitis C, B, HPV, EBV, etc. But they define out those oncogenic viruses, and really SARS-CoV-2, the causative agent for COVID-19, it really matches every box there. But then also long COVID and the mechanisms it does of reactivation of oncogenic viruses, chronic inflammation, signaling pathways, all of this, but it ties right into what you're doing, but it ties into what I'm doing at the Williams Cancer, but it ties into something you posted recently, and you've been great on Substack. By the way, where can they find you on Substack?
Oh, yeah, it's called Decision Junction. So if you go on Substack and you type in Decision Junction, that's my Substack. And then I'm also on X, so I do quite a lot of X posting as well. And they can see me there under Beyond Bio. And I can provide those links for you.
Yeah, great. And I'll ask you again at the end as well, but please do. And we'll put those in the show notes. So check out the show notes. As always, you're going to see all the supporting evidence in the show notes. So please check those out. You posted an article, and it was really touching in on the off-target toxicity of the messenger RNA. We really have a tunnel of thought thinking about how is the messenger RNA affecting us now. But I think we're really missing the boat of where we're going with this. And you posted an article, and the whole topic of the article was the protein-peptide collapse. But I'm getting down here to the article. It was published in in Nature magazine, and it was entitled 2024, N1 methyl pseudouridylation of messenger RNA causes plus one ribosomal frame shifting. You have to truly be a nerd like me and John here, Dr. Catanzaro, to see this article and read it and just go, I want to read it again. So this gets into the underlying mechanisms of what this messenger RNA is doing. So John, your expertise, both in that engineering mindset, but, Boeing, that is a complex process. I mean, building a jet, an engine that flies a jet that carries hundreds of people across the globe, that's no simple task, number one. Number two, taking care of patients with cancer, that's an equally complex task. And now you're in the biochemistry, the precision care. So all three of those, I think, bring you to a focus point to actually be a great expert to touch on this article, but touch on the potentials about messenger RNA and what are the implications, not just where are we going, turbo cancer, et cetera, but how. What exactly is happening? And a good place to probably start is what the heck is protein-peptide collapse?
Yeah, protein-peptide collapse happens when the reliable platform for encoding the functional proteins of our body are in error. So it is known as transcription errors, right? So when we talk about ribosomal shifting, it simply means that all of a sudden there's a knock-in that creates a really serious bad hiccup in the transcription rate and in the codon and the actual codon encoding displays that you find that build these proteins. Now, with the mRNA technology, because the mRNA technology is kind of, I consider it flawed in a lot of ways because it's, number one, premature. It's very erratic. It produces way too many rogue, unpredictable effects, right? So when we look at it, here's the thing. We know that the viral aspects, things that happened during COVID-19 were synthetically derived. And we also know that the mRNA activity for the vaccination platform or the vaccine technology was also synthetically derived. So you have actual synthetic elements that are now introduced into the cellular machinery that are foreign. And because they are foreign, the cellular machinery then becomes aberrant in its activity. Aberrant simply means faulty patterns that are not part of the normal expression between the communication from the nucleus to the endoplasmic reticulum, which is your transcription factory, right? To which your ribosomes are part of, and the actual end product development that the cells depend upon for function, which are the proteins, right?
And I think
when we have that type of glitch, go ahead.
No,
Yes.
wait, it's really important because you're getting into some highly biochemical technical talk. And, I love this, but for the listeners out there, understand that our genetic code, our genome, it is fixed. It takes thousands and thousands of years to change one aspect of that. But what Dr. Catanzaro here is talking about is the process by which our genetic code, our genome, is transcribed into RNA, messenger RNA. And so when you talk about the messenger RNA vaccine, and I've always said, when you look at COVID, it's whether injection via or infection, that spike protein is a toxin. I think everybody misses that messenger RNA of the spike protein is a toxin. But from that messenger RNA, it gets translated into a protein. So when he was talking about proteomics, he was looking at the end component of that critical aspect of where the genes are being transcribed into RNA and then being translated into actually protons. And the codons he's talking about is how that process, which is an amazing, beautiful process, how these proteins are actually put together by amino acid. So I just wanted to put that in there, John. So please take over.
Oh, yeah. Now, and if you're looking at this, you're looking at some 37 plus trillion cells in the body, right? So when we're talking about system compartments, we're talking about different system compartments, the neurological system, the cardio system, the immune system, all of the microbiome, all of these programmed elements in our body are continuously being remodeled by normal cellular machinery processes that allow for proper conversions, proper development of cells. And when you actually have what we call a synthetic element like a spike, which then creates more rogue protein development, then you have these functional proteins no longer become functional. They actually become seriously impaired. So when we talk about peptide protein collapse, it's those individual peptide sequences that make up the protein, and that collapse is what causes what we call a molecular debris field, if you will. It no longer becomes a functional tissue. It becomes a fibrotic mess. It becomes a molecular debris field. It's like a massive explosion occurs because of the fact that we actually see this synthetic element that creates such a chaotic disturbance, right? And we're seeing this on molecular expressions of patients' data. So we have a collaboration with the University of North Texas Genomics and Bioinformatics, where we are actually validating the detection of this spike from the synthetic virus and from the synthetic mRNA elements, right?
So either infection or infection.
Yeah.
Okay.
Right. Infection or inoculation, right?
Yeah.
And then what we're doing is we're looking for that cross-embedding, action that's happening back into the DNA to create a genetic instability that creates more bad messaging coming from the mother, the mother molecule, which is the DNA, which then creates that corrupt activity from the cellular reproduction, because it's actually saying, okay, I want you to type this message, and I want you to continue to type this message because the DNA now has it encrypted in its own matrix, right? So the thing is that that's bad. That's not good. That's bad. That genetic instability. And it's not that we're trying to create alarm here. What we're trying to do is we're trying to say, look, we have to understand this process. Why do turbo cancers turn into turbo cancers? Well, if you want to describe it in scientific terms, what basically it means is that it's an aggressive cellular proliferation and migration that is actually uncontrolled and severely dysregulated process to where there's no brakes to be put on it, right? And the thing is that when you see that happening, the resistance of that process goes up higher and higher and higher with new generations that are cells are being produced with that bad encrypted message, right? Yep. So as well as I do, for during the whole process of COVID, everybody was saying is that there's no possible way that, the current vaccine strategy using the mRNA technology was going to allow for a DNA to be corrupted, right? Because it's not going to happen. It's not going to be a retro action back into DNA. Well, I got news for you. That's not true.
And
it's
now,
not true.
There's actually literature being published on that, though, because, a lot of people are talking about the contaminants of DNA in the vaccinations,
right.
But we're actually talking about, again, messenger RNA. It's that middle step from the genetics to the genome to the protein. And it's that encoding of information, because that's really what the messenger RNA, and it's being retrograded. It's being retro applied back into the genetic code. So it's RNA going back into the genetic code. Now, the end result there is when we look at cancer, people go, well, let's go back to the somatic theory of cancer, which is cancer is a genetic defect. Well, there's some minor truths to that, but what we're understanding there, that it's a much more complex issue, and that the genetic defects, they arise out of a lot of other dysfunction that occurs with it, mitochondrial, metabolic, inflammatory, etc. But if you have a patient that has cancer, this is where I think that turbo cancer phrase really applies, is you're instituting a genetic, as you said, debris field in a process where in many ways it already exists, because what that's going to do, the genetic, the biochemical molecular pathways, which cancer so disrupts, here it further disrupts it and further pushes these cells out of homeostasis. And as you talked about the normal cellular pathways that kind of clean up bad cells and then, create new cells, it just further throws a monkey wrench into this process. But that's exactly what's happening. Is that a decent evaluation of this process?
Yes, that's a very good way of describing it. And when you're looking at these specifics, see, like when we're talking about, there's some cancers that don't have an inherent genetic instability to them. They have what they have environmental causes from extrinsic reasons, and that may not necessarily have, an inherent genetic instability with them. However, when we actually have an outside force that's coming in to create a very a missignaled environment, like for instance, what we know controls certain type of events for cellular reproduction, then we actually see, okay, here are some of the pathways that are, like for instance, the W&T pathway, the Notch pathways, the hedgehog pathways, the DNA mismatch repair pathways. These just to name a few. And then they're related crosstalk sister related genes that are responsible for communication back and forth. So when you have a lot of that aberrant activity, it's a massive windfall, because it creates that effect on the collateral pathways that are controlling the homeostasis environment, where the homeostatic mechanisms become severely altered, severe dysregulation, failure to recognize where the balance point is. The cells are totally disoriented. The transcription is terrible, and it's just a bad result, right? So understanding that environment is quite complex. You have to understand that, yes, I mean, in cancers, we know that, that's the reason why it's so easy. Like if you have direct kill happening with chemotherapies or radiation, or take the offending thing out of the body. We understand the significance of all that, but bad signaling, it doesn't go away automatically. It just doesn't shift away from and create a new norm. Sometimes those compensatory mechanisms, especially when we're starting to see this mRNA, mRNA disruption occurring, it allows for propagation. So it allows for recreation. It allows for what we call reproducing. It becomes then a prime mover for manufacturing of its own kind. And the thing is that we see that you have synthetic spike with a message from DNA coming back to create that message from the nucleus, going back to the transcription system to say, create more spike, create more. It's just like when we are infected with a virus, and the virus goes in, the cell becomes a manufacturing outlet, and it pumps out more of the same virus that creates the infection in the first place, right? Well, it's the same thing that happens with this spike protein. With this development of spike, the manufacturing happens, the cell becomes a manufacturing machine, and it produces more unrest in the cell, and it spits it out into the extracellular matrix for further manufacturing by other cells, right? So it's like, it's a cycle that continues to aggressively replicate in the billions, billions.
That's a good point, because cancer thrives on mutability. The faster it metabolically grows, the more mutations it spits out. The analogy I give is, you're driving down the road, you blow both tires, axles on the concrete. The faster you go, the more sparks you spit off. It's that, quote unquote tumor mutational burden. So the more mutability, the more adaptability it has to evade the immune system, grow and spread. And so what you're describing here is a mutability that's increased exponentially because of this synthetic messenger RNA.
Yes. Yes.
Now, I've
Yeah.
proposed that the impact on our individual bodies is one thing, because I think that even those of us that have not been vaccinated, we've been still exposed, whether that's we got the infection or, shedding from others. But what I'm concerned about, and I really don't hear anybody talking about this, and I'd love to get your take on this, John, because you have that kind of engineering mind, but you had that clinical experience, is the legacy, not just in our cells. So turbo cancer is something we see clearly today. But what about generations to come? What about our children? What about my grandchildren? I have a grandkid now, almost 4 months old, a little boy, looking here. So,
Right.
there's something called transgenerational inheritance of pathology, whereby epigenetic changes happen, and these are inheritable. But we're actually talking about something that's actually being incorporated into DNA. Do you think this is going to be something that's going to have an effect in legacy, meaning in our offspring, or is it something that's going to be confined to this intra-generation?
No, I think it's going to have some transgenerational impacts, and we're going to see that, unfortunately. And so therefore we're going to have a much more aggressive posturing ourselves to create a mitigation and clearance. We're going to have to, without harming the further. In other words, we're not going to go in there and try to program a bunch of genes through splicing and things like that. That's not going to fix the problem. What we need to do is we need to come up with a mitigation clearance strategy that allows for the proper protection mechanisms to occur now, being that this has been introduced into the environment and is being replicated in the population. So the thing is, it's very important that we begin to understand that it's going to take a much more aggressive innovation for us to be able to provide a corrective path, right? And it's not going to be through vaccination. It's going to have to be through, more personalized approaches where you understand what's happening on the person's own molecular symmetry, what their signaling is. Because here's the thing is that we've already seen in the literature that, people respond to different ways in different systems. Their neurological systems will have neurovascular CVAs, or they'll have a very bad myocarditis, or they will have aggressive organ failure and breakdown of tissue. Where, we probably you've heard of the glioblastoma case of the one gal who's, very prolific out there as a journalist, and she winds up coming down with a massive glioblastoma very shortly after she was vaccinated with the vaccine. Well, it happens that fast. You see, that's the thing, is it happens that fast, and the effects will continue to be replicated, through the generational influences. So the thing is, I really do believe that we have to collectively be thinking on an engineering, like you said, the engineering mindset for innovation to be able to come up with a very effective mitigation strategy, and allowing the body to make these corrections to protect transgenerational effects, right? And if we can actually get something in place to where there's a good intervention, especially when it comes to molecular editing, from a standpoint of the editing for function, we could definitely slow down, if in fact halt a lot of these very serious complications that are happening.
Because when we look at the effects of this, peptide protein disaster, I love the verbiage you said there, just a debris field. I love that. And so think about the inside of your cells just being a debris field. There's some immediate effects, the toxic aggregates, the chronic inflammation that results, and then the immune dysfunction, misregulation. And understand that is prominent in autoimmune disease. The immune system attacks, other parts of the body because it evades, it doesn't, identify self as self. It starts to target the self.
Yeah.
And when you look at cancer, almost all of our cancer patients have some degree, whether they're diagnosed or not, of autoimmune disease. And in many ways, I kind of look at cancer a little bit, John, as somewhat of a autoimmune issue because the immune system gets turned on the immune system. Of course, it's the cancer that's doing that, but there's some autoimmune characteristics of here. And so when we talk about immune system dysregulation and misrecognition, you can see how this process in acute phase in a cancer could actually flare it. But here's the long-term implication that I think has you at the frontier of this and really gets into the legacy aspect of it. It's the long-term disease process of cancer. You mentioned neurodegenerative disease, cardiovascular disease. I mean, you can't watch the news in a day or two and not hear about some suspicious person that drops from a cardiovascular event, whether that's causative at that point or not, autoimmune disease and organ failure. You've kind of touched on every one of those, but and I didn't really give you my notes ahead of time. So that's exactly what the literature points to as a long-term process, not just in the individuals, us, our generation, but it's our grandkids yet to even come. And so, but that's why it puts Neo7 Bioscience right there in the front. How does precision cancer care, or whether it's autoimmune, whether it's long COVID, neurodegenerative disease, as you mentioned, how are you positioned as a company in precision peptides? How are you positioned to actually provide a resolution, a new problem, new solutions through precision peptides?
Yeah. When you're talking about N of one engineering, right? And a lot of people, when they talk about N of one, they just think, well, you're not going to be able to mass produce that. It's not scalable, blah, blah, blah. That's not true. That's all false. The N of one engineering is essential in conditions like these, because the thing is that you're not going to have the same expressions in people and treat them as a mass. Here's the thing with vaccination is everybody was treated alike. And so it's a mass population vaccination. Well, people's bodies don't work that way. They don't work that way. And then when they're given a vaccine, they're not going to have the same effects. Person A is not going to have the same effect as person B. Person C may have a real bad, accelerated bad effects. And here's the thing about this is that when we're talking about trying to create this means and motion for dealing with this hijacked system, if you will, you have to realize that the body becomes disoriented. Identify friend or foe is no longer in great effect. So we have in the military, in the Air Force, pilots always had the IFF acronym. Identify friend or foe, right? And that would be the directive to where their kill shots would be, right? Because they're identifying friend or foe. And in the body, when the body is disoriented and it's not able to identify friend or foe for whatever reason, the HLA system, which is the system that controls the central movement of being able to have a proper surveillance and adaptation, susceptibility, compatibility, how well you're going to your systems are going to posture themselves for protection, all based upon that HLA expression. Very important. If it's hijacked, it becomes a mess, right? To where the immune system doesn't have any directives. It has no directives whatsoever to intelligently mount up a fight, right? So you get to start seeing, not that we want to go into these conspiracy things, but you can see how it becomes more along the lines of a weaponry that is not, neutralizing the defenses of our systems. And the thing about that is it's a very subtle and sly process. It's not something that's as, easily tangible to see. That's why I say that it has an unpredictable component to it, and it's going to be very difficult to create the right responses to that unpredictable action, right? So that's the reason why these N of one designs are so important, because you're going to find out what the molecular expressions are at the core in that individual. So that means that the fix is not going to be a commonized fix. It is going to be reparative mechanisms dynamic to the person's expression of how that fault created that whole change, and how you're going to be able to mitigate and clear that process by a better effect, functional effect coming in, right?
Yeah.
That's more in alignment simply with the body.
Yeah,
Right.
N of one literally means there's no one like you. And so thus this therapy should be operated underneath that premise that this therapy needs to be unique to your molecular nature. You know, if you sit in a clinic and you're 6'6", 360 pounds, you're sitting next to a 5'2", 125 pound, and you go, why are they getting the same dose of medicine I am? I have a different cancer than they do. I'm male and she's female. What gives? Everybody intuitively knows this process of N of one, but medicine is slow to adapt to it. So, but that precision nature of this, I think, is where you're situated to provide these solutions in cancer, neurodegenerative disease, long COVID, and autoimmune disease. And honestly, I think these are the big areas that we are seeing changes in from an epidemiologic perspective. But I think we're going to see this in the future. And this is where you come in as a delivery of a strategy of a therapy that actually promotes regeneration, reversal, healing. And by the way, that's what it means to be a physician, healer. So it really ties into that, but it's not a one size fits all. As you said, it's that N of one. It's that, okay, here are your precision peptides, 6'6", 360 pounds, 5'2", 125. Here are your precision peptides. And they are different.
Yeah. So, you land on a very important point, even in conventional standards. So if conventional standards were more in alignment, like let's use, Opdivo or Keytruda as an example. If you were to precision personalize the dosing to where it's more sensitive to the person's own molecular expression, and like, for instance, personalized therapeutics that we do sensitize the system for those conventional immunotherapy agents to work much more efficiently. But if you actually created a dosing structure that's more dynamic to the patient's own molecular, makeup, where you don't give them a blasting dose, you give them maybe a third of that dose to create a better effect, or you go local like you're doing, in the work that you're doing, to where you hit the local aspects of it instead of blasting the system with it. I mean, it makes more sense because it's a navigational movement that's more dynamic to the patient's own molecular symmetry, right? And of course, you're naturally going to reduce the off-target toxicities because you're keeping that in mind, and patients are going to tolerate it better. And they're also going to have a better response too.
That's
right. I mean, you get to see that in higher percentages of patients when you start tailoring those type of regimens that way. Patients have much better toleration, but they also have much better sensitivity, less likely to develop a resistance to the agent, right? Because that's a common problem out there, developing resistance to the agent that's employed. And they'll have much better outcome in a lot of cases.
So three areas that I'm hearing, obviously, as it relates to cancer. And again, I think you got to throw autoimmune disease into that. And then that opens up a whole another door of, diseases, neurodegenerative and everything else. But it's, as you mentioned, increasing the sensitization. So, increasing the ability of a therapeutic to actually be effective. Now that's looking at things through a systemic chemosensitization. That's something that a lot of therapeutics are look to do. How can we improve the efficacy of this drug being delivered to a target and actually elicit the effect that is intended? Obviously, the Williams cancer, we go into the tumor. So that's one of the ways we bypass some of the limitation of the off-target toxicity in that whole delivery method. But here we're talking about just increasing the sensitivity overall, but the ability to actually reduce the resistant capability, which is obviously a big effect in cancer treatment, is when cancer becomes resistant through that highly mutability state, Now you start to run out of therapeutic options. And when cancer patients run out of therapeutic options from their doctor, that's where things really get grave. But what about two other areas? What about regeneration? Here, I'm talking particularly about regeneration of the immune system, because, it's really interesting when you look at the literature, John, is that, cancer is a disease of the aged, and people think that's right. Older people have cancer. Well, that no longer applies. I mean, I talked to a 26-year-old with stage 4 cancer from Canada about a month ago. That's not an old person. That person's my age. Goodness gracious. She looked like a child. Stage 4 breast cancer, bone mets. But the immune system, I think, is the key there. What about when cancer, the aging of cancer cells? When I'm talking about that connection, it's actually that cells are aged, meaning the immune system cells operate as if they're much, much, much older. And when you look at chemotherapy, radiation, even surgery, those further accelerate the aging. So a double negative doesn't make a positive. Do you think that the precision peptides here have the ability to reverse that aging trend and actually regenerate a younger immune cell, or a cell that functions at a younger state?
They have the capability of creating that revitalization. So because they are, programmed elements of, working on stabilizing the functional activity of the proteins of function. And what we see is we do see the regenerative effects, right? So you see the programming effects going in to stop the excessive migration, metastatic proliferation, and some of these resistance characteristics that you see in cancer hallmarks. Or even you can see the similar effects, like you were saying, with the aging and even neurodegenerative disease, where it has almost the same characteristics of functional loss, and the gain of that activity is very slow. So what the peptides do is the peptides create a moiety of an opportunity of propagation. So you go in there, you're creating that functional stability on that protein path. Then that message is caught, and it's actually stabilized in the cellular matrix, and then it's reproduced by other cells developing with that stronger representation. And then before you know it, you have a better tissue viability. Your tissues around recover. They get better. They get healthier. They become more resilient, more robust. They have a better immune defense strategy themselves, because they're no longer dysregulated, right? They're actually coming up with a clean signaling path. And that's the whole idea about peptide programming, is it's a natural mimicry that allows for better programming effects. And it doesn't happen lickety split. It happens over a segment of time. So usually what we see within a 6-month period, like for instance, we have this muscular dystrophy kid that we designed for it. He's been on these personalized therapeutic that we designed for him for 4 months. We just got a call, last week where the doctor said, I'm going to tell you that this kid made a total 180-degree turnaround. She said a 360 from dysfunction to actual having functional gain that he's never had. So this is a very difficult neuromuscular type of a disease process, to where, and what did we do? We looked at his multi-signal platform that was aberrant, resistant, and unique to his expression, right? And we saw that it was very faulty. So we created those engineering elements that allowed for creating that functional gain, improvement to occur for revitalization and regeneration.
And then
anyway,
when you look at the extrapolation of this, obviously for that individual, it's significant. For the sarcoma patients you talked about in remission, that's significant. But we're talking about when you're regenerating, when you're changing the playing field, when you're changing the environment, when you're empowering the immune system, building the defenses here, you're actually working in a prevention strategy, both for that individual, so intragenerational, but then transgenerational for generations to come. So it's actually a proactive therapeutic strategy for the individual for future disease. Take for example, long COVID, and really the unknown future of what we understand for those patients. We have some understanding of what the immediate is, but we don't have, 10 years, 20 years of what is going to be the manifestation of this last 4 to 5 year experimentation. We have to be honest, because that's exactly what it was. It was an experimentation. What are going to be the implications of that? I think we're only beginning, John, to seeing just the tip of the iceberg of that, but you're really able to help on these multiple fronts through this precision peptide therapy. And understand these peptides, the peptides are the building blocks of proteins. And you may go, I've never heard of a peptide. What do you even talk about? Let me drop a name that you may have heard of. Insulin. That's right. Insulin peptide. Right. So these are very real in biological function and from a pharmacological function, right? So this is very real, strong, deep, solid science that John Catanzaro is talking about. So John, what's the future bringing for Neo7 Bioscience?
Well, Neo7 Bioscience is, working to continue to develop the personalized engineering strategies. Our science team, our bioinformatics team, we're also in a collaboration with the University of North Texas. As I mentioned earlier, the Genomics and Bioinformatics and their Life Sciences Institute. So we're in a lot of different developments for innovation. So like, in other words, how it's going to benefit the individual and how we can actually solve these complex problems, right? And we've been having a lot of inquiries everywhere. And we're excited about these inquiries because we are creating more engineering innovations, in these related areas of active disease, these long COVID problems, what's called turbo cancer. I mean, we are in it. We're in the thick of it, and we're working to create resolution and action for hopeful solutions, right? A lot of people talk about the problem, but we're focusing in on what are we going to do about it? And we have to really focus in on it. You actually mentioned something about sarcoma, which I thought was very interesting. The sarcoma, like healing sarcoma, you know that about the EWS fusion, right? Oh, yeah. Everybody in the research environment is making a big thing about trying to focus in on the fusion itself, where very few people understand the significance of the collateral damage that's created by the fusion. And if you actually look at the fusion found in healing sarcoma, targeting the fusion is unproductive because it already happened, and not every sarcoma cell expresses it. Right. So you're going to have some sarcoma cells that are they're going to be negative fusion in the same person, and you're going to have some that would express fusion. And it all depends upon the process of development, like where the cells are in their maturity index and how, like you said, the mutable effects of it, right? So you get to see that targeting the fusion is not the way to go, but targeting the collateral signal pathways that are creating downstream bad effects are the way to go. That's where that multi-target ability comes in. That's the reason why we have success in our healing sarcoma patients, because we are not focusing in on what the institutions are saying, focusing on the fusion. It's not really the effective way to go. You focus on the collateral damage that the fusion causes. So that the body has the accommodation ability to fix those bad signal pathways. And that, again, that's a propagated effect. And those peptides, that's where peptides come in this mimicry environment. They're not just about stimulating the immune system and the T cells and the B cells and all this stuff. Everybody talks about them as fashionable neoantigens. They're not just antigens. They're not, that's false labeling. What I say is that they are dynamic editing features that allow for the body to make constructive changes that are very beneficial and that can have profound influence over a long period of time. So if we're really going to have a transgenerational effect, this platform and getting it to perfect even more is probably one of the best ways to go for this innovation, to have a long-lasting transgenerational positive outcomes for
Yeah, I
very
agree.
complicated problems.
I agree. Well, John, thanks for joining us. As always, it's been a deep dive down the rabbit hole of biochemistry. I love the conversations with you. Where again can the viewers and the listeners find you and find out more information about Neo7 Bioscience?
Well, the website is Neo7 Bioscience.com. My X page is Beyond Bio. And I'll text you the link to that. And then a Substack page is Decision Junction. I can get that to you as well. Those are the three best ways to track the work and get a hold of us and communicate to us.
Well, John, keep up the good work, and obviously you're a dear friend. Keep up, let's keep up the communication. Definitely want to have you back on again. Hopefully we can connect in person soon. I want to thank everybody for joining us on this episode of Prescribed by Dr. Nathan Goodyear. You can find out more information by going over to pre-scribe.com. I ask you to please help us spread the news of this innovation and other just innovators out there that are working together to provide the solutions of the future, but today for the complex problems that sometimes just are unforced errors. Download the podcast, share, like, and subscribe. Encourage you, please give us a 5-star review, and we will see you on the next Prescribed by Dr. Nathan Goodyear podcast. Thanks for joining us on this episode of the Prescribed Podcast. All references supporting this episode can be found on the prescribe.com website. That's pre-scribe.com. To get your next dose of medical innovation, make sure you subscribe to Prescribe. We'll see you for your next refill.